Microfluidic droplet networks represent a fascinating frontier in science, where liquid droplets, each acting as a miniaturized compartment, are interconnected to form programmable systems. These networks aren't just random clusters of droplets. They're structured like a circuit board made of liquid, capable of performing precise functions, useful in chemistry, biology, medicine, and even computing. But how can a network of tiny droplets, each sometimes only a few microns across, be controlled and made to work together? It starts with the unique properties of droplets in microfluidic environments. Inside narrow channels, droplets can be precisely generated, moved, split, merged, or stored. These droplets behave like individual cells in a biological tissue or like logic units in a computer. By arranging them in a sequencer grid and controlling their interactions, researchers can build networks that mimic neural circuits or chemical reaction pathways. One striking example, researchers have created droplet arrays where each droplet contains a specific reagent, enzyme, or cell. When needed, the droplets can be fused together to initiate reactions on demand, almost like telling parts of a biological system when and where to activate. This is particularly useful for high-throughput screening and drug discovery. Instead of running one reaction at a time in a large beaker, thousands of reactions can be tested simultaneously in a droplet network occupying just a few square centimeters. Another powerful application lies in synthetic biology. What if you could build a synthetic tissue out of water droplets suspended in oil, where each droplet is a tiny bioreactor or sensor? Scientists have done exactly that, using droplets that communicate through chemical gradients or embedded nanopores. They can simulate signaling like that found in biological cells, allowing for the study of cell communication, artificial tissue engineering, or even smart drug delivery systems. Why do droplets need to form networks instead of staying isolated? Because networking allows complexity. Think about neurons. One neuron is useful, but a network of them gives rise to thought. Similarly, droplets in a network can perform operations over time and space, sequential chemical reactions, conditional logic operations, or coordinated biological functions. Some systems even mimic logic gates, using droplet interactions to build liquid computers. A challenging question arises: how do you ensure stability and prevent cross-contamination in such small, dynamic systems? That's where surface chemistry and channel design become crucial. Surfactants are often added to stabilize the droplets and prevent them from sticking together unintentionally. Microfluidic channels can be patterned to guide droplets precisely and to ensure isolation between different paths. Researchers even use electric or magnetic fields to steer droplets in programmable ways. More futuristic developments include droplet-based machine learning, where droplets serve as data carriers or processing units. Another emerging area is droplet robotics, where autonomous systems use droplet logic to perform tasks like detecting toxins or navigating chemical gradients. Microfluidic droplet networks represent a blend of fluid dynamics, chemistry, information processing, and biological mimicry. As these systems become more refined, we may see networks that function like adaptive synthetic organs, real-time diagnostics on a chip, or programmable materials that respond to their environment. In a world increasingly reliant on miniaturization and integration, could droplets be the next platform for computing diagnostics and bio-inspired engineering? The growing field of droplet networks suggests the answer could be yes. Microfluidic droplet systems generate discrete volumes of fluid within immiscible carrier fluids, typically oil and water phases. These droplets act as isolated microreactors, preventing cross-contamination and enabling high-throughput analysis. Surface tension, flow rates, and microchannel geometry govern droplet size and stability. The ability to produce monodispersed droplets underlies many analytical and synthetic applications. A T-junction chip forms uniform water and oil droplets for PCR reactions. Like forming bubbles in a soapy stream, each droplet is a self-contained compartment. Droplet networks form when multiple droplets are brought into contact and interconnected through lipid bilayers, electric fields, or embedded channels. These connections allow for controlled communication between droplets via diffusion or design pathways. Networks can be dynamically reconfigured to model complex systems or perform logic operations. This architecture mimics biological tissues and neural circuits at the microscale. A microfluidic chip connects 10 aqueous droplets using lipid bilayers to study ion transport. Like neurons connected by synapses, droplets pass signals through designed interface. Droplet behavior in networks is governed by flow rates, surfactant chemistry, temperature, and applied fields, electric, magnetic, acoustic. These inputs modulate droplet fusion, division, sorting, or content exchange. Precise control enables programmable fluidic operations similar to logic gates or digital processors. Feedback mechanisms are often integrated to maintain desired functionality. Electrowetting is used to merge selected droplets for on-demand chemical reactions, like controlling traffic at intersections using lights. Input parameters govern droplet decisions. Droplet networks serve as microreactors for parallel or sequential chemical synthesis, reducing reagent use and improving reaction control. The confinement within droplets allows for rapid mixing and reaction kinetics due to high surface area to volume ratios. Complex reaction cascades can be orchestrated by merging droplets in a defined sequence. This modularity facilitates combinatorial chemistry and drug screening, creating a library of polymer nanoparticles by sequentially fusing reagent-containing droplets. Like baking individual cupcakes in a muffin tray, each droplet hosts a unique reaction. Droplet networks replicate compartmentalization found in living cells, making them ideal for single-cell analysis, artificial tissue construction, and synthetic biology. Each droplet can contain cells, DNA, or proteins, enabling reactions such as cell-free expression or enzymatic cascades. Droplet connections can simulate cellular communication through molecular diffusion or vesicle fusion. This allows modeling of organoids and early embryonic development. Linked droplets are used to express proteins in separate chambers and study signaling between them. Like organs in a body exchanging hormones, droplets coordinate biological functions through connections. Droplet networks can be embedded with fluorescent, electrochemical, or enzymatic sensors to monitor biochemical events. Each droplet functions as an independent sensing unit or part of a sensor array. Signal readouts can be collected optically or electronically and correlated with spatial arrangement. This makes them powerful tools for point-of-care diagnostics and environmental monitoring. A droplet network detects glucose concentration gradients using embedded enzyme sensors. Like pixels in a camera sensor, each droplet contributes to the overall data picture. Droplet networks can implement computational logic by controlling droplet flow, presence, or content. Boolean operations are achieved via fusion, blocking, or splitting at microchannel junctions. Fluidic systems thus act as analog or digital circuits, useful for autonomous decision-making in diagnostics or smart devices. Droplet logic can operate without electronics, using only fluid dynamics. A network performs an AND operation by merging two droplets and detecting a signal only if both are present. Like mechanical computers using gears and levers, droplet networks compute with fluid. Droplet networks are used to assemble materials such as gels, colloidal crystals, or structured polymers with precise microscale architecture. The droplets act as molds or delivery vehicles for building blocks like nanoparticles or biomolecules. By controlling fusion and flow, materials with spatially resolved chemical or mechanical properties can be fabricated. This approach supports next-generation soft robotics, responsive textiles, and biointerfaces. A droplet network assembles a patterned hydrogel for soft tissue engineering. Like 3D printing with liquid pixels, droplets lay down structured materials. Future droplet networks will integrate real-time sensing, feedback control, and machine learning for autonomous operation. Hybrid systems combining optical, electronic, and biochemical interfaces will enhance droplet-based decision-making and therapeutic delivery. Integration with lab-on-a-chip and organ-on-chip technologies will expand applications in diagnostics, personalized medicine, and artificial intelligence. The convergence of droplet microfluidics with synthetic biology and nanotech promises transformative innovations, An intelligent chip routes drug-loaded droplets based on patient biomarkers detected on site. Like a miniature brain, droplet networks will think, sense, and act at the micro scale,